Tobacco Mosaic virus or TMV. The first ever virus that was discovered was tobacco mosaic virus. TMV was first discovered on tobacco, hence its name became tobacco mosaic virus. This virus can cause infections in the tobacco leaves, and here we can see that some spots on the leaves, these mosaic like structures, and here also the green coloration is disappeared and mosaic like structure we can see on the entire leaf. That's why, due to the symptoms which it's caused in the leaves, mosaic like symptoms, so this virus is called mosaic virus. And since it was first observed in the tobacco plant, so it was named tobacco mosaic virus. But this virus is not only limited to the tobacco but many other members. So this is the structure of the virus. So in this presentation we will see some basic properties, structural properties of tobacco mosaic virus. First we will see the history of this virus. In 1892, the Russian botanist Ivanovsky demonstrated that an extract from the infected tobacco plant could transmit disease to healthy tobacco plants after passages through the Chamberlain porcelain ultrafilters. These filters were known to trap most of the bacteria. Filter are fitted, porcelain filter are there, so these filters can trap all the bacteria. But the extract which he obtained from the tobacco leaves, from the infected tobacco leaf in fact, so that extract can cause infection, can redevelop infection in the healthy tobacco leaves. So something was there in the leaf extract of those tobacco plant that cannot be filtered through these porcelain ultrafilters, but it can cross the filters. It is beyond the pore size of the filter and it can cause the infection. So that was something interesting. But however, Ivanovsky didn't understand the full significance of his findings. Later on, Dutch microbiologist Martinus Beijerinck replicated Ivanovsky's experiment and he got the same result. And on the basis of the finding of the experiment, he given the term viruses. So the Beijerinck also known one of the founding father of virology. Viruses, which he called contagium vivium fluidum, means a contagious living fluid, a fluid that contain the pathogens that can be infectious to the healthy one. However, Beijerinck's vision of viruses was not appreciated or confirmed during his lifetime. Later on, Rosalind Franklin, who was a British chemist and X-ray crystallographer, she hypothesized that TMV particle was hollow and its RNA genome was single-stranded. Due to his scientific work on the viruses and the structure of DNA, she managed to get 2 Nobel Prizes. Now we will see some structural features of TMV. So this is the diagram of tobacco mosaic virus, and these are the actual images of tobacco mosaic virus. So here we can see so many rod-like structures. So this is the actual structure of tobacco mosaic virus under the electron microscope. So its structure is very simple. This virus has only nucleic acid and a protein coat that is called the capsid. So first property is TMV particles are helical shaped. So you can see a helical RNA structure, so they are the helical. The dimension of this virus is, it is 18 nanometer in diameter and around 300 nanometer in length. So these are very narrow, 18 nanometer in diameter and 300 nanometer long. Its capsid, the outer covering, it contains 2130 identical CP subunit. So here we can see these are the identical protein subunits, and if we count them, the number is 2130. These are the coat protein subunit or the CP subunit. This virus contains a single molecule of positive sense single stranded RNA. So this red color structure is positive single stranded RNA, and its length is 6400 to 6600 nucleotides. The protein and RNA ratio of this virus is 95 to 5%, means this virus has 95% protein and only 5% of RNA. TMV virions can self-assemble. In other words, purified TMV RNA and coat protein will self-assemble into stable rod-like helical virus particles. The virus self-assembly process takes little free energy to occur. Means if you provide a RNA molecule of this virus and coat protein, so the coat proteins will automatically, or they will self-assemble around the RNA molecule and they will form a virus particle. TMV is one of the most stable virus, so it is very difficult to destroy this virus. It is resistant almost to all chemicals and many physical agents which generally deactivate other viruses. The coat protein of this virus is extraordinary resistance to most proteases, which are the enzymes which degrade the proteins. Now the host range and how this virus transmit. So TMV virus is a broad range virus. It infects almost 550 species of different flowering plants. It can infect tobacco species, peppers, tomatoes and potatoes. So the most common host for this plant virus are tobacco and other plants of the Solanaceae family. So this is the family Solanaceae, so which is the target of this virus. This virus can easily be transmitted from one crop to another by the mechanical transmission, or its debris or its particles are there in the roots and soil of infected plants. However, this virus is not known to be spread by insect vector transmission. So mechanical transmission is the major mode of the transmission. It is very stable virus and it can remain for 2 years in the soil if soil is not exposed to the freezing and drying. So they remain alive about 2 years in the soil. They are stable in plant residues in soil and in the compost of tomato plants. So compost is also not safe. So tobacco mosaic is one of the most persistent disease of tomatoes because the virus can remain infectious without a host for many years and it able to survive high temperature. How we can limit the spread of this virus? We should use uncontaminated soils for the production of seedlings. We prohibit smoking during the work because the tobacco which is present in the cigarettes may be a source of this virus in the fields. So in the fields smoking is not allowed. Workers should wash their hand regularly so that if the virus is attached on their hand, so mechanically it cannot be transferred to the healthy plants. We have to remove and destroy all the infected or suspected plants from the nurseries which are infected with TMV. We can spray the plants with skim milk or buttermilk. So these substances can change the pH of the leaf surface, which can inactivate the virus particles. We have to destroy all the weed source of TMV in tobacco and nearby field. We should rotate crops every 3 years with maize and corn, avoiding tomato and pepper crop because they are the host for the virus. We should use TMV resistant varieties for the cultivation. So by this practice we can limit the spread of TMV.